Transport across cell membranes. You need to know the fluid-mosaic model of membrane structure, and before we define what that means, we're going to take a look at the components of the cell membrane. Most importantly, we have the phospholipid bilayer. This is the arrangement of the phospholipids into two layers, where the hydrophobic fatty acid tails get repelled by water, so orientate to the inside of the membrane, whereas the hydrophilic phosphate head are attracted to water, so they orientate to the aqueous environment either side of the membrane. Embedded in the phospholipid bilayer is proteins. These proteins might be intrinsic or extrinsic. Examples of intrinsic proteins are channel proteins, which form pores in the membranes, or carrier proteins. There may also be extrinsic proteins, which are found in one layer of the phospholipid bilayer, whereas intrinsic span across. And an example of this is a glycoprotein or glycolipids. Glycoproteins are just proteins with polysaccharide chains attached, while glycolipids are lipids with polysaccharide chains attached. We also may have cholesterol, which binds to the phospholipid hydrophobic fatty acid tails. So why do we call this the fluid-mosaic model? Fluid just means that molecules within the membrane can move laterally, for example the phospholipids, and mosaic just means that it's a mixture of phospholipids, proteins, glycoproteins, glycolipids, and everything else. The fluid-mosaic model of membrane structure can explain how molecules can enter and leave a cell. So we're going to link each of the components to methods of transport. So the phospholipid bilayer allows the movement of non-polar, small or lipid-soluble molecules, for example, oxygen, water, and these move down a concentration gradient. This is simple diffusion. It also restricts the movement of larger or polar molecules. However, we do need to transport some large or polar molecules across the membrane, for example glucose. So to do this, we have channel proteins and carrier proteins. These facilitate the diffusion of water-soluble or polar molecules or ions or large molecules, and this is called facilitated diffusion. Again, it's moving down a concentration gradient. In addition, carrier proteins allow the movement of molecules against a concentration gradient, and this uses energy from ATP. This is called active transport. There are features of the cell surface membrane that also adapt it for its other functions. So, for example, the fact that the phospholipid bilayer is fluid means that it can bend to take up different shapes, for example during phagocytosis, or to make vesicles. Also, the fact that we have some surface proteins, for example, glycoproteins, these often act as receptors or antigens and therefore have a role in cell communication and cell signalling. The role of cholesterol is to regulate the fluidity and increase the stability of the membrane. It does this by binding to the fatty acid tails, causing them to pack more closely together, so it restricts the lateral movement of the molecules, making up the membrane, making it more rigid. We can split our types of transport across cell membranes into two categories. First, we have passive transport. These don't require energy from ATP, and the reason they don't need energy from ATP is because they utilise a concentration gradient, or in the case of water moving, we say a water potential gradient. We've already discussed what simple diffusion is, and briefly what facilitated diffusion is, but just a few other things to mention is that these carrier and channel proteins do facilitate the movement of specific substances. You need to know the role of these, and the carrier proteins transports large molecules. The protein changes shape when the molecule attaches, and the channel proteins transport charged or polar molecules. They act as a pore. Some of these can be gated so they can open or close. Then we have osmosis, which is the movement of water, and we don't say that it moves down a concentration gradient. This time we say it moves down a water potential gradient, and this is across a selectively permeable membrane. A dilute solution has a high water potential, and a more concentrated solution has a low water potential. This just relates to the likelihood or the potential of water molecules to move out or into a solution. So pure water would have the highest water potential, which is actually still just 0, and adding any solutes to a solution lowers the water potential and makes it more negative. I have just realised as well that I've written the wrong thing on here. It should be the other way around here. So more sugar, less water would be a more concentrated solution with a low water potential, and vice versa. With all of these things as well, all of these types of transport, I should mention that these are all the net movement. So things, particles, molecules can move both ways, but it's the overall movement. In contrast to passive transport, we also have active transport. This type of transport requires energy from ATP, adenosine triphosphate, and therefore this allows molecules to be moved against a concentration gradient, so from low to high concentration. It does require the use of carrier proteins, just like we saw in facilitated diffusion. Different carrier proteins do actively transport different specific molecules. And the ATP specifically is used, it's hydrolyzed to release energy, and that changes the shape of the tertiary structure of that carrier protein to push substances through. So I'm just drawing for you what that would look like. So we can see here that a molecule would bind to it. So for example, this might be glucose is being actively transported. A molecule binds to it, ATP is hydrolyzed, that changes the shape of it, and that pushes it through. Something else that you need to know about is cotransport, where we transport two molecules through one carrier protein, which we would call a cotransporter protein. Now, the reason I've put this under active transport is because this carrier protein might use ATP, use the energy from that itself to push these two molecules through in the same direction or opposite directions. For example, the sodium-potassium pump, which we'll look at in a moment. Or it could indirectly be active by the sodium-potassium pump or something else. Another mechanism might use ATP in order to generate some sort of concentration gradient, so that there's a concentration gradient of one molecule, which can therefore, as it goes through its cotransporter protein, will pull another molecule with it against its concentration gradient. Now that might sound a little bit confusing, so we are going to look at an example. All you need to know really about this is the example, which is the absorption of sodium ions and glucose by cells lining the mammalian ileum, which is part of the small intestine. So what I'm drawing here is an epithelial cell. On one side of it, we have the lumen of the ileum, and on the other side, we have a capillary. Now, we are trying to absorb glucose from the lumen into the epithelial cell and therefore into the capillary. So we're going to have a really high concentration of glucose in the epithelial cell so that it can move down a concentration gradient into the capillary. So firstly then, how do we get this glucose from a low concentration in the lumen to the epithelial cell? Well, we're going to use a carrier protein. We could just actively transport glucose, but we make things a little bit more complicated. Instead, what we do is we use a sodium-glucose cotransporter protein, and this basically utilises the fact that there's a concentration gradient of sodium, where there's more sodium in the lumen of the ileum and less in the epithelial cell. So sodium naturally moves down its concentration gradient into the epithelial cell, and sort of with it, glucose jumps on and it also goes through. So the energy from this is not directly provided by ATP, but it's provided by the gradient of sodium. But actually, weirdly, this is sort of active transport, because the way that we get that concentration gradient of sodium in the first place is by actively transporting sodium out of the epithelial cell into the capillary. This is another type of cotransport. We don't just transport sodium on its own. To keep all the ions balanced, we also transport potassium inside, and this is called the sodium-potassium pump. So this is there to create a concentration gradient of glucose. So all of this works together to create a high concentration of glucose in the epithelial cell, and therefore it can move down its concentration gradient by facilitated diffusion from the epithelial cell into the capillary. And this would just be through a glucose channel protein. Remember, a channel protein that's specific to the movement of glucose. So really this is quite a complicated process, But there are two examples of cotransport in this. We've seen the type of cotransport where it doesn't directly need ATP, but it's where two things move and we're kind of utilising the concentration gradient of one thing, in this case sodium, to move something else against its concentration gradient. We've also seen the example of cotransport where we're transporting two things in opposite directions. This is the sodium-potassium pump. And this is there to set up a concentration gradient. You need to be able to use your knowledge of transport across cell membranes to decide on factors affecting the rate of transport and therefore what sort of adaptations cells would have to increase their rate of transport. So the first thing is the concentration gradient or a water potential gradient in the case of simple diffusion, facilitated diffusion and osmosis. An example of how we adapt to maintain a high concentration gradient is when we're trying to absorb oxygen into the blood and we're trying to remove CO2 from the blood between the alveoli and the capillaries. We have a very vast blood supply from our capillary network. This is also seen in the small intestine. And because the blood is constantly moving due to circulation, we're constantly bringing away the blood, for example, in the around the alveoli with lots of oxygen in, with a high concentration of oxygen, and bringing back fresh blood with low oxygen concentration in, which maintains this concentration gradient. If the blood wasn't moving, all the oxygen would move in, and then it would just reach an equilibrium and no more would move. Another thing that affects the rate of diffusion and osmosis is the diffusion distance. So, for example, we have thin capillaries, which are often an exchange surface. These are 1 cell thick. Another thing is the surface area. So a high surface area increases the rate of any sort of transport. So, for example, the microvilli, the extensions of the epithelial cells, create a high surface area. Now, one more thing that would be specific to facilitated diffusion would be the number of channel or carrier proteins, or in the case of active transport would just be carrier proteins. In these cases, increasing concentration doesn't increase the rate of movement, facilitated diffusion, to unlimited extents. It can actually get limited by the number of carrier or channel proteins. So the increase in the rate will stop increasing once all of these proteins are in use, or we could say that they're saturated. These types of transport, because they rely on carrier and channel proteins, they're also affected by the speed of these proteins, the temperature they're working at, and the pH, and because these all affect the tertiary structure. Another thing with active transport is the amount of ATP. ATP is produced by respiration, so the rate of respiration would also therefore affect the rate of this transport. I've seen quite a lot of questions where you're given where they give some cells a respiratory inhibitor which stops respiration, and you're asked to explain the effect of this on active transport, and it would stop it because we don't have any ATP to transport it against the concentration gradient. There are 2 required practicals in the transport across cell membranes topic. The first one is to do with osmosis. It's where we try and determine the water potential of plant tissue. What we do to do this is we create solutions with known concentrations of a solute, for example sucrose, and therefore we know their water potentials. And what we do is we measure the effect of these solutions on the mass of a plant tissue. So, for example, little cylinders of potatoes. We pop them in there and we see what happens to their mass, which shows us whether water is entering them or leaving them. So if the mass increases, that shows that they're gaining water by osmosis, which therefore shows that the solution must have a higher water potential, because water moves by osmosis from an area of high to low water potential. The opposite, if they decrease in mass, that means they've lost water by osmosis, so the solution must have a lower water potential. What we're looking for is when the potatoes or the plant tissue stays the same mass after spending a long time in these solutions. This means that no osmosis is happening, no net movement, and therefore the solution must have the same water potential as that plant tissue, for example, as the potato. So a little bit more detail then, technical details about how we do these things. So we call these series of solutions with different concentrations a dilution series. You need to be able to use the maths to know how to calculate how to dilute something to get a certain volume of a solution when we're using like a stock solution with a high concentration, and how to dilute it to a particular concentration. So I'm just going to give you an example here. So we've been given a 1 mole per decimeters cubed solution of a sucrose solution, and we want to use this to make a series of different concentrations. So we've been told that we want to get 30 centimeters cubed of a 0.015 moles per decimeters cubed solution. So we're trying to dilute this. So the formula that we can use to do this is we divide our required concentration by the concentration of the stock, and then we multiply that by the volume required. So in this case, we want 0.15 moles per decimeters cubed. Our stock is 1, and then we multiply that by 30. That tells us how much of that stock solution we need to use, and then we're going to just top the rest up with some distilled water. So we want 4.5 centimeters cubed of 1 mole per decimeters cubed sucrose, and then we add the rest is distilled water. So we add 25.5 centimeters cubed of distilled water. Some other technical details that questions commonly ask you about is how you do it and why you do certain things and what you could control. So when we're getting our plant tissue, what we want to make sure we do is that we're cutting equal sizes or length of that tissue. So for example, the potato, we could use a cork borer and cut them to the same length. We also want to control some other things, such as using the same tissue, using the same potato, for example. What we're then going to do once we've got these cuttings of our plant tissue, our potato for example, we want to measure the initial mass and then place them in the solutions. We're going to leave them for some length of time. Now, questions often either ask you, why do we leave them for a long time? That is to enable them to equilibrate. So all of the water that's going to move in or out is going to move out. So we're actually seeing by the end of it, not it sort of halfway done. Why we would keep temperature constant throughout. So we would keep that constant because that's going to affect the rate of movement. And how we would do that is we would have a water bath at say 30 degrees, and what we would do is we would measure the temperature as we go with a digital thermometer or something, and then every time it sort of fluctuates, which it might, especially if we're having it at room temperature, then we would sort of counteract that. Another thing we do once we've taken our potatoes out, or whatever plant tissue we're using, carrots or something, we want to blot them dry with a tissue briefly before we reweigh them, because the amount of water on the outside of the cylinders that they've picked up might vary, and this will affect the mass. So we actually only want to see the mass of it itself, not any mass of water that's on the outside of that. So then what we're going to do is we've got a table. We would have our different concentrations of sucrose or whatever solute they've been in. We've got their initial mass, their final mass, rather than just calculating how much different they are, because they might all have slightly different initial masses, and so that we can compare with other results, we're going to calculate a percentage change in mass. Percentage change is just change divided by original. So this enables comparison. As with any sort of practical, we want to do repeats at every concentration here, which will enable us to identify anomalies, reduce the effect of them, and it's going to make the mean more reliable. So I know that's a lot there so far, but just remember, so far all we've done is we've just measured the mass of different potatoes, how much they've increased or decreased mass. But what we're trying to do is we're trying to find the solution in which the potato doesn't change mass, okay? And we might not have an exact one. We've done a range of values. So the way that we can find when there's 0% change in mass is to plot a calibration curve. We do this with the percentage change in mass on the y-axis, the concentration or the water potential on the x-axis, and we plot our points, and this is called calibration curve. We draw a line of best fit, and therefore we're going to find the concentration where the curve crosses the x-axis, where there's been no change in water, so no net movement by osmosis. So we know what concentration it is, and this is going to correlate to some sort of water potential. So we use some sort of table or resource to find the water potential of that particular sucrose solution or whatever. Our next practical then is looking at the factors affecting the permeability of cell surface membranes. So two of the factors that do affect permeability are temperature and pH. For example, the concentration of ethanol. These could be the things that we change, therefore these would be our independent variables. How we look at the permeability of cell surface membranes is we're going to use some sort of plant tissue that contains coloured pigments. For example, a beetroot, which contains a dark purple sort of pigments in the vacuole. And as these things change, we'll see the more permeable it gets, the more these pigments will leak into the surrounding solution. Therefore, a more intense colour shows that there's a higher permeability. We can measure this quantitatively using a colorimeter or semi-quantitatively, just comparing to a standard. Things that we would control in this experiment is we make sure that we keep the source of the plant tissue the same, we keep them in the solution for the same length of time, we use the same size or shape of plant tissue, and it's also important that we keep the volume of water the same so that we can compare our results. If you had too much water, then the concentration might appear lower than expected. It's important when we're changing something like temperature that we're monitoring it as we go, whether that's a thing that we're controlling, if say we're measuring, if we're seeing the effect of pH, the concentration of ethanol, then we want to keep the temperature constant, or whether it's a thing that we're changing it. So we could take readings throughout using a thermometer or a temperature sensor and adjust accordingly. So the results that we would get from using a colorimeter, the more light that passes through the solution means the less concentrated it is, and therefore the less permeable the cell surface membrane is. The way that these things would actually affect the permeability is temperature would increasing temperature increases the fluidity of the phospholipid bilayer, and above a certain temperature it might also denature the transport proteins, whereas pH, increasing concentrations of ethanol, might actually dissolve the bilayer and it also might denature the proteins.